Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, we would like to take a moment to thank our ecosystem partners for their support, namely Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. Please mark your calendars for our next Dr. GPCR symposium held on Friday, September 22nd on the topic of GPCRs as therapeutic targets. The symposium is free, but you must be a Dr. GPCR Ecosystem member, which is also free. Please see the menu button for the symposia page at the top of our welcome page to check out the program and register as a presenter. You don't have to register as an attendee. You just have to be part of the Dr. GPCR Ecosystem to get access to the links to join us. We also have set aside 2 hours for networking and poster presentation on Kumo Space that day. The posters don't have to be fully formatted. Just come with a few slides and have a chat with your community members about your favorite GPCR or other signaling proteins that you're working on. If you'd like to present during your work during the networking time on September 22nd, please complete the submission form available on our website. There is no limit on the number of posters we can accommodate. To present, you'll only need to provide us with the title of your presentation, a 1-minute video abstract, and come with a few slides to help you discuss your work with your colleagues. Everyone is welcome. If you have any questions or comments, or if you'd like to present your work but don't know how to join us, please feel free to email us at hello@drgpcr.com. What are you doing between November 2nd and 4th? Join me and the Dr. GPCR team at the 22nd edition of the Great Lakes GPCR Retreat at Chateau Montebello in Quebec. We at Dr. GPCR are proud to be supporting this iconic meeting. I've had a blast help organizing this meeting this year with my colleagues from the University of Ottawa, the University of Montreal, and the University of Sherbrooke. Visit gpcrretreat.org for more details on the program. The early bird registration and poster submission deadline is September 8th, and the late registration deadline is September 21st. Hurry up and join us between November 2nd and 4th. And now let's dive into this episode. Good morning, everybody. This is Yamina from Dr. GPCR, and this morning I have with me Dr. Françoise Bachelerie. Françoise, welcome. Very excited to have you. It was a great pleasure to meet you finally in person at the Gordon Conference last June. So, and let's start at the beginning. Would you please introduce yourself?
So, my name is Françoise Bachelerie. I'm French, as you can hear with my accent, French accent. So I'm French, and I have done most of my scientific work in France, and introducing to which point do you want me to introduce?
When did you get into science? What do you work on? I think it's kind of a given that it has to do something with chemokine receptors, as you and I were. Actually, you were one of the authors on my very first author paper.
Yeah, so I have indeed obtained a PhD in the 90s. And my PhD was on the regulation of the well-known HIV virus. And at this stage, I was working in a lab where we have some interest in the regulation of the transcription of HIV. And later on, during my PhD, we have been in contact with people working on this crazy receptor, which are chemokine receptor, and we didn't know it was eventually important for HIV to interact with those receptors. And we have been in the field thanks to those guys, working with them together on HIV on our part and chemokine receptor on their part. And we found, together with other people in the field, that indeed chemokine receptor and CXCR4 was the main co-receptor for HIV to entry into a cell. So we knew at this time that HIV was interacting with CD4, but we were, broadly, looking after this entry receptor. You remember that? You don't remember, you are too young, but it's a story. So and we find out that the chemokine ligand of CXCR4 that other people has found as an important way of entry of HIV this chemokine was indeed a real, true natural inhibitor of entry of HIV because it was competing with the envelope. So this was my first encounter, if I can say so, with GPCR through the chemokine receptor, which at this time in the 90s were not very well known. And thanks to this first observation that was really followed quickly by another observation that CCR5 was another chemokine receptor for the entry of other type of HIV. So the field just explodes, the field of chemokine receptor, but really explodes. And it was discovered many, not so many, in fact, it's a small family of the class A GPCR. And so this domain has been pushed a lot thanks to this HIV viruses. And we were thinking at this time that it was the key to find some inhibitor for HIV entry. Unfortunately, HIV, as you know, is a virus which is mutating a lot. So it was just a very good idea, but in the fact it was not possible to use these approaches. And we know now that it's still difficult to target a GPCR, even from other family, you know. And definitively, so CXCR4 has been highly investigated thanks to that. And we know today that this receptor, together with CXCR12 and these atypical receptors that we have been discovering in 2005, is really a trio which is necessary for life and very important for many aspects of the homeostasis of human, but also, you know, pathology. Of course, they are hijacked by many pathological processes. So that's how I enter into the GPCR field.
From an HIV.
In the 90s, yeah.
And then, but before you did your PhD, you know, in virology and working on HIV, did you always know you wanted to be a scientist? I'm thinking here, you know, as a teenager maybe, or as a child.
I was not aware of that until my mother discovered a letter of intent from me when I was 14, and indeed, I was already claiming that I want to be a scientist, but I forgot about that. And this commitment. So eventually I succeed in that, at least.
It's, you know, sometimes our guests, sometimes they say, yes, I always knew I loved science. Others say, oh, I had no idea what I wanted to do. And it's so interesting. I think you're one of the few first guests who say that as a very young age knew that they wanted to be a scientist and actually grew up to become scientists.
Yeah.
Were you more of a chemistry or more of a biology, you know, student?
Actually, I was more, when I was at the university, more prone to do some stuff in the development. I'm relatively old, so it was the beginning of the development with Drosophila, and you know, it was really interesting to do genetic at this stage, at this period of science. And unfortunately, I was not allowed to go in this direction, so I moved to biology, you know, and I am doing my master degree in Pasteur Institute, you know, because I was hired there. And it was just a good choice as well, because it was just a great time for biologists, you know. HIV was a period really exciting for us.
Wow.
Really sad, but really exciting as
well. From a science perspective. And it's interesting because you mentioned, you know, CXCR4 and CCR5 being discovered as HIV co-receptors in the 90s. I know it's 2023, but when I think about it, it's not in my mind. Maybe I'm getting old as well, but in my mind, the 90s wasn't that far out, and it's incredible how far we've come since then. How
Yeah.
much more do we know about these chemokine receptors, and even HIV? So when you finished your PhD, you mentioned that your career has been in France. Have you ever moved out of France for a postdoc or?
Yeah, I have been doing a postdoc after my PhD at UCLA,
Mhm.
working still on HIV, and I spent there 3 years, 3 very nice years, working in Chen labs, and it was really very nice period of my life, very constructive, interesting, and motivating, really. And then I came back to France when I tried to get a position, a permanent position, which is a way of doing science in France. You
Mhm.
need to have this permanent position, which is not easy to get. So you have like competition for that. So, but again, my the time I spent at UCLA was just wonderful. Yeah, I really
It,
enjoyed. Yeah.
it's interesting, you really went to the other side of the world to do that. How did you pick the lab?
It has been easy. I think it's still easy for a French PhD to get a position in US. It's not so difficult.
Mhm.
Um, for I don't know. Main reason is maybe because we are still relatively well-trained PhD students, so US like it.
Yeah.
Europe, European student in general, and also because in US it's more flexible to get money, at least for some period of time, and after that the postdoc gets its own money, which was already the question at this time. So
Yeah.
back again to the decision, it's because I have I mean my friend at this time was already at UCLA, and I want to join him. That's all.
Mhm.
I tried to find a lab who interests me in
Mhm.
the field of HIV.
Yeah.
And the field of HIV was very, very, um, what can I say, dynamic, really
Yeah.
dynamic. It was relatively easy again to find a position there. So it was quick in a few months
Wow.
in the site. So, and I moved after my defense.
Yeah. Wow. How was adapting from the French culture to the, LA culture? Did you
Oh,
find it
very easy for me. I find it very easy. I like it very much, the LA culture. It's
nice?
really cool, really cool, really
Yeah.
cool.
Yeah.
And we were living in a part of the city which was also very cool. It was in
Mhm.
West, so it was nice. And I like it a lot, very much. So that's the good part. The other part of being, adapt um, to the PI of the lab, it was not so easy. He was you know, used to behave in a way that I was not used to with
Mhm.
postdoc and PhD students. So
Yeah.
it was a little bit tough for me, that part. But he went slowly within, nevertheless. He went smoothly, but slowly,
Good.
smoothly within, despite the
Mhm.
fact that we were very different. I was
Yeah.
not used to be directed like that. So
Mhm.
it's easy for me. And it's funny because during this part of my life, I have been in contact, thanks to my the PI of the lab, by people doing movie, you know. So
Wow.
I have some experience in movie as well. It's
Well,
really, yeah,
that's what happens when you move to LA.
apparently, yeah, it's a very classical way of, So I have been doing that, and it was funny, very funny experience.
Can you, and without really putting any pressure, can you share anything funny, a funny story or anything that happened to you in LA while other than being in the lab? You mentioned movies. I'm
Yeah,
really
it's
curious.
very, very funny time because it's a movie on HIV, and a very nice movie done in order to tell the story of HIV and how the AIDS communicate among people, the society and scientists as well, and what was the fight between French team Montagnier and Barré-Sinussi on one side and the other side the bad guy in US.
Who
Which
shall
stolen French story, French virus. So no, a nice movie, a funny movie. It's really regarding that story because the PI of the lab tell me that he has been contacted by a director of movie in order to do this movie, an HIV. And a special part was supposed to be in Pasteur Institute, so they took a mansion close to Beverly Hills where the kitchen were looking almost as, a laboratory in Pasteur with,
wow.
wood and white wall and stuff like that. And because I was from Pasteur, he decided to ask me to go there, and I have been there. And it was really so funny because the guy is a prep master, who decide what he can put on the stage. Made a mistake. So he put all the apparatus were from dentists,
Oh, yeah.
Nothing was scientific at all. It was so funny. So yeah, I told him nicely, no, you made a mistake. It's not the good one. So very funny.
So were
And
you in the movie, or you just gave advice as to how the lab should look? And
so I gave advice first, and unfortunately it was important advice, necessary advice for this part.
yeah.
And then because I was playing the game, the guy decided to hire me for more, sophisticated parts, which were uh, the translation for French from English to French, because they were having some French actor, well-known French actor, and they want to come there in order for them to either speak in French and be translated in English or do both. And when the crew arrived, the French crew of actor, I was there still because he asked me to go there to be there. So in order to train, French actor to behave as scientists, to get some.
Wow.
It was so fun. So really, really fun.
Well, I think you just made the top one, ranked number one, because I think that you are the only guest on the podcast who was in the situation.
So to answer your question, yes, you can find my aunt in the movie because
Wow.
the lady, the actress, which was supposed to be Françoise Barré-Sinussi, so the Nobel Prize, didn't succeed in really having the tube in her hand, or it was a mess. So I said, okay, this is your hand, and we will have her face on the movie.
Wow. So you have
Yeah.
your hands. Oh, wow. Do you remember the title of the movie? I have to look it up now. And we have to link it somewhere.
And the band played on.
Okay.
The band played on. Yeah.
I'll look it up. This is so cool.
Really cool.
What an experience. Who would have known that you went for a postdoc and you ended up working, well, helping out with the movie
Yeah.
while
And
doing this.
sometimes obviously with them because it was so funny. I like it very much.
Of course. Wow. That is amazing. So wow. I'm in awe. This is why I love the podcast
And
because
another funny thing in this story is that we have been to the butcher with a master prep guy in order to find some lymph node,
uh-huh.
We were looking at some pieces of food in order to look like a, lymph node from a guy
Yeah.
we are supposed to decease because of the AIDS, It was
Wow.
really funny. Anyway.
Wow. Wow. See, this is why I love the podcast. You would never have the opportunity to talk about experience making movies at a conference because this is just so unique.
That's so interesting.
I think it is. It just doesn't may not have the space in a talk about about chemokine receptors and
Yeah,
HIV.
yeah,
That is amazing. So then so after your postdoc, you come back to France and you start your own group
yeah.
and you continued working on HIV and then chemokine receptors and the family. But how can you tell us briefly how your scientific interest evolved with time?
Yeah, I can. So I was really focused on HIV, as you understood, from a very long period of time. Until we have been discovering when I was back in France. So, oh, I'm sorry, I forgot to cut the
No
sound.
worries.
When I was back to France, I have been involved in a story indirectly where some people. So I told you that CXR4 was my main interest. Nevertheless, the chemokine field was really my interest, and those three partner of the CXR12, signaling axis
Yeah.
are completely required for life. So not so many mutation has been reported, almost none,
Yeah.
except in the 2000, early 2000, where some geneticians told us that they were having people most likely having mutation in
Except
CXR4
for.
that were reported by a group, American group, a very bright American group of people uh, making the association between a polymorphism and a mutation in CXR4, heterozygote mutation, and a very special syndrome, with some immunodeficiency, I mean, deep immunodeficiency and a susceptibility to develop human papillomavirus infection. I mean, not infection, but
Mhm.
pathology, And in France, at the same moment, those type of patients were found close by hospital close by Paris, and they join us and we decide to move on this topic, which was really interesting because it's always more interesting, especially now that we know that science should be focused on the more and more, which is not, which is understandable, but not always a good choice. But anyway, we should be focused on pathology, and try to find something related to concrete situation. Why not?
Yeah.
But in this case, it's always better, to my point of view, to start with the disease and try to find some molecular
Yeah.
key.
Yeah.
So we were here facing a situation where those people have a mutation,
Yeah.
most likely, causing the disease, but we were not completely sure of that yet. And with some, apparently, some selectivity toward the development of pathogenesis for one viruses, not all viruses, one viruses. So despite the fact that those people were deeply immunodeficient, they were living
Yeah.
relatively well up to 40, 50, with difficulty, but still, they were facing well their pathology, and they were having this special, susceptibility toward HPV pathogenesis. So HPV, it's a viruses, how viruses. We know that we have almost 400 type of viruses, HPV viruses today, which we know from the early 2010, so less than 10 years ago, that they are part of the commensal, microbiota. So
Mhm.
they were well known to be pathogen, causing some cancer, I mean,
Yeah.
cervix cancer for at least 80 88% of cervix cancer are because of those HPV.
Mm-hmm.
The same for oropharyngeal cancer, you know.
Yeah. Yeah.
And they are suspect to be responsible as well for non-melanomal skin cancer, you know, suspect, not sure. And despite all this bad pattern, you know, we know from early this year, very, very recently, that they are part of the commensal microbiota. And even they are the most important eukaryotic viruses on the skin. So meaning that you and me are carrying, without any lesion due to HPV, no warts, nothing, we are carrying between 8 to 11 different type of HPV. And it's not, you know, it's not known and it's not seen because HPV, it's hijacking the skin differentiation process, you know, and because of that, HPV is taking advantage of that to go with the desquamation, you know, and it's really transparent for the immune responses. So that's why I have been shifting a lot to this type of interest, which was really different. Viruses are very different. So sometimes it's not well known when we are outside the field, but working on one virus doesn't mean that you know everything on every virus. So it's really difficult to jump from one to another one with original idea. And also at this time in the 2005 and those type of years, we discover with other that this field of atypical receptor, you know, chemokine receptors that are not apparently signaling through G protein, and they have another way of signaling, and they are most likely regulating the functioning of normal classical, you know, GPCR. So it's really important. I move a lot from my previous study.
Yeah, I have to say, I always found your papers very elegant.
Oh.
I really, really, really enjoyed them, and not because we were co-authors on one, but I really, since I wrote my thesis on chemokine receptors, and there was CXCR4, CXCR7, or ICKR3 and CCR2, I had to go through a lot of papers, and I always thought your papers were very elegant.
Oh, thanks.
I was wondering, so this is something that I've always, I think the pathology we're talking about is the WHIM syndrome with the mutation in CXCR4. But when you talk about, you know, CXCL12 and the other two chemokine receptors, which are CXCR4 and CXCR7, what I'm wondering, it is known that CXCR7 also interacts with CXCL11. And has anyone ever looked at, you know, this kind of a not necessarily trio, but include CXCL11 in there, and what does it do?
Yeah. Of course, many people try to be exhaustive in this way. It never ends in a way because you behind interacting with chemokine ligands, those two receptors, ACKR3 or CXCR4, are also, I mean, presumably interacting with natural ligands which are not chemokine, you know.
Yeah.
So it could be, it's difficult to pretend to be exhaustive and looking at CXCL12 and CXCL11 at the same time, and, you know, at ACKR3 and CXCR4. You know, and I saw in your list of questions that you were trying to ask us what we could emphasize as a difficulty in the field. I mean, most likely this is it, you know, because you cannot pretend to be, you know, to handle everything in a correct fashion. I mean, especially because it's really flippy to work on ACKR3. It's a receptor which is internally, I mean, most likely intracytoplasmic, not at the membrane. So it's really difficult to look at it carefully in a non-invasive way, you know. So, I mean, it's relatively tough. So yeah, it's a good idea to have a global view on the quatuor or whatever you want to incorporate in the
Yeah,
study. It's really very ambitious sometimes.
it's very difficult. Yeah, I always wondered because there's papers that show that CXCR4 and CXCR7 form heterodimers just to complicate things a little bit more. And then they both bind CXCL12, and then there's CXCR3, which has different splice variants. And I've always wondered, well, what do those splice variants do on their own, but do they heterodimerize with CXCR7? Considering that they share, I mean, I think this is the beauty of the chemokine field, is that there's so many, there's 20 something receptors, 50 plus ligands, and then everybody could be interacting with everybody.
Yeah.
And it's difficult because in an organ or in a tissue or in an organism, all of these receptors and chemokines can be expressed, and you cannot, it's hard to do an experiment where you have 3, 4, 5 receptors in there and then wonder what happens. One of the big questions that I always got during my thesis is, what would happen if you mix the chemokines? And I said, wait a minute, we're not there yet. We can barely look at one-on-one ratio between receptor and chemokine, but that's also something complicating things a lot.
It's really it's definitely something we would like to reach, all of them, all of us,
Yeah.
That it's a really complicated story and question that nature knows how to address, apparently, because when you have many chemokine on the matrix, extracellular matrix, which are sitting on heparan sulfate with a very, very high affinity, we are close to the nanomolar range as well. So they are there sitting there, and it's really difficult to modelize, indeed, to model. In this type of environment, how one receptor or homodimer or heterodimer or whatever can make that choice.
Exactly.
Because we are speaking about receptor which are involved, most of them, in the migration of the cell. So the cell is not sitting there, the cell is moving as well. So you have also to take into consideration the dynamic of the situation and the extraction, from the endothelium to the tissue as well. So imagine all the contact we might need to model in. We are speaking about eventually model, organoid or whatever. It's really amazing how interesting it is, and complicated at the same time. So and on one way, when you try to model in mice, it's nice because you have all the environment, but it's mice, and it's tough to have molecular point of view, access to molecular events. And when you try to model in vitro, it's tough for reducing, so you have to the middle between all of that.
You take what you get. Well, which is also a good thing in the sense that there is much more work to do. So there's much more opportunity for us to make discoveries as a whole scientific community in that sense. So let me ask you this: what is your favorite GPCR?
My favorite one.
Yes.
It's maybe still CXCR4.
Yeah. That was my first receptor I worked on as well.
Yeah, I know.
So it has a special place in my heart as well.
It's good.
No, I think it's a very interesting receptor, and in our common publication, I started that work making those mutants of the dry motif during a summer internship in Nicolaus's lab. And interestingly, I think it took 3 months to get those 6 poor mutants, because back in the day we were using the Kunkel methodology where we're making single-stranded DNA, and we didn't even have ATP in the lab. The lab was just starting out. So I have a very very strong feelings around making those mutants, and it was so nice to get the sequencing data back, which is also, I didn't have to run sequencing gels, but still it took, I think, a week back in the day to get the sequencing data back, and you had to take the tubes and walk and hope that you get what you need.
Yeah.
So we talked a little, well, we talked a lot about chemokine receptors and viruses. And we talked also about the difficulty in the field, considering the complexity of everything. If you could, what are the tools that you think we need? Anything that would help us really dissect the different chemokine and chemokine receptor pairs, and also put them together. And you can be as, wishful as you want. I had someone working on adhesion GPCRs one day, and the person said, it was Anthony Boucard, and he said, well, I wish I could like become this tiny and then go into the cell and observe adhesion GPCRs activate. So for the chemokine field, any thoughts, as any wishes putting out there, what kind of tools we need?
I think it's a very nice wishes that you just mentioned, and it's great, really great. I would be the same, a little things to go on the cells and travel with the cells.
Yeah.
In the blood,
Yeah.
and into the skin, where I like the skin, and try to encounter some papillomaviruses eventually, and find out how does it work with them,
Yeah.
So yeah, I think it would be nice to travel.
I think so too. I think so too. There was this cartoon back in the day about the magic bus, the teacher and the magic bus, and they would shrink and go into these different things. I think something similar to that would be really phenomenal to go in there.
Sort of scary as well, because this big synapse, immunological synapse, eventually, engulfing everyone.
Yes. I think so too. But I can only imagine the number of events happening every minute in a cell. And yeah.
Imagine that, because the cell is moving, it's completely deformed,
Yeah. Yeah.
You have the little age, you have everything which is, she need to face a lot of difficulty.
Yeah. And then you have the shear stress in the bloodstream, and things are moving fast. It's just, I think it's fascinating.
Yeah, definitively.
So
awesome. Yes.
Okay, let's do it. Let's find the magic bus then. I always ask this as well, and the answer is always yes. But do you think GPCRs are still good drug targets? Now, if you could elaborate a little bit about that.
I'm not sure I catch the question.
So do you think GPCRs are still good drug targets?
Yeah, I don't have to think about it, just to look at what's going on, and that's the pain is really important. The depression is very important. And obviously for me, all the immunological and infectious disease, so I mean, obviously we have to face the reality. GPCR everywhere.
Yeah, they do everything. And it's interesting that, I sometimes when I'm thinking here about the keynote at the Gordon conference from Fiona Marshall, where she was presenting, well, it wasn't only about GPCRs. Obviously, it was focused on the GPCRs. And when we talk about the complexity of systems such as the chemokine system, I'm always fascinated at the fact that we were able to so far develop the current drugs.
Yeah.
Because it's like playing the lottery. Obviously, very directed and scientifically, sound, but still.
Yeah,
wow.
Still, I understand your point. Yeah, I agree. Yeah.
It's just, we love our GPCRs.
Because we can manage a specific effect so far in a tube, but what about the reality when it's in the body? It's another story. Yeah, I agree.
No, agreed. All right, last segment of our conversation. What would be, in your opinion, your top 3 aha moments that shaped your trajectory as a scientist?
If I understood well the question, I believe it could be scientific or it could be
Both. Anything you want. Anything that changed your trajectory. Something that happened to you that, shifted you from whatever you were doing before.
yeah. So obviously it was the travel to of the Atlantic, to go to
Yeah.
UCLA, it shaped definitively differently my mind and my
Mhm.
wishes and everything. And and uh, and and yes, I mean, despite the fact that I was working on HIV, that was so important, a very important pandemic, the first one we really have faced so
Yeah.
far before the COVID. I was not really, in the field of medicine at all. I was really doing academic research. So the WIM story shifted me a lot on the medical field. And this is why, and thanks to that, that I have been in contact with medical and MD, PhD, and patient.
Yeah.
So that has been a big field, big move for me, a big, really one big. So, and I think it's the two most important three. What would be the third one?
I think the third one, based on our conversation, could be something around the fact when chemokine receptors were identified as HIV co-receptors. I think that
Obviously,
that
and it's really amazing how viruses are, intimately related to those infectious bug. And,
yeah,
that's really amazing. Yeah, yeah,
yeah,
it's really a great domain, biology, and I will really push young people to go there. It's not the future, but in a way, sadly speaking, it's the future as well because we have to face
yeah,
so many,
yeah,
so trouble with those viruses. The viruses are, the next one on the line.
yeah. Speaking of advice to junior scientists, what would be your advice to
Be
scientists?
virologist.
Be a virologist. Stop, drop everything, be a virologist.
working on GPCR, obviously.
Yeah, obviously.
Obviously. Yeah.
But anything else, I think one of the things that came through the conversation is the fact that you went from France to the other end of the world to do a postdoc, and it allowed you to really shift and mature your way of thinking about science. And I think that's important advice for anyone thinking about the postdoc.
Absolutely. It's make you curious and also um, in a way free of more free of thinking. I think so because you are not belonging anymore to one single culture or whatever. You need
Yeah,
to address those type of challenge in your life. That's absolutely mandatory. I agree with you. And also, yeah, doing this job is being at the beginning highly motivated, you know,
yeah,
to ensure that you can afford frustration and because you have some higher perspective or longer perspective,
yeah,
longer
yeah,
view. So
yeah,
it's really important. Not everyone today can afford this type of frustration. I think so. It's not the way of acting in or actually
yeah.
around. Yeah.
No, I think so too. I think if you have a long-term goal or long-term vision, which is bigger than yourself and bigger than just quote unquote publishing papers, like in your case, you mentioned really the patient-driven approach and the focus on GPCRs, viruses in the context of patients. I think that's you can take on the frustrations of the daily things better, thinking about, okay, this is the goal. This is just a bump in the road.
Yeah, yeah, exactly.
Well, on that note, Françoise, merci beaucoup. Thank you
Yeah.
for your time today. I really enjoyed our conversation. And I'll talk to you soon. Don't go anywhere. We're gonna let people wonder what we talk about after we stop recording.
Okay, thanks for the invitation, Emilia. Thanks a lot.
Thank you. Thank you for joining us and listening to this Dr. GPCR podcast episode. We would like to thank our Dr. GPCR podcast guest and our team members, Attila, Ines, Montserrat, Ivana, Andreina, Balint, and Julia. Please mark your calendars for upcoming 2023 Dr. GPCR Symposia. Don't forget to visit us and check out the program. A huge thank you to our ecosystem partners for their support: Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. You can connect with our partners directly in the ecosystem. Join us today. Also, please subscribe to the Dr. GPCR newsletter. Find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. Another great way to support us is to share your favorite Dr. GPCR program with your network and colleagues. With any questions or suggestions, please email us at hello@drgpcr.com. Until next time, stay safe.